like to start by thanking the organisers for the opportunity to present my research here. So breast cancer, as we all know, kills by metastasizing. And it's been found that primary tumours begin shedding neoplastic cells into the circulation at an early stage, and this shedding is independent of tumour size. Once they've emerged, once they've arrived at distant sites and begun to grow, tumour metastases evolve independently. And it's found that circulating tumour cells in the peripheral blood may represent the earliest cells with metastatic capability. And if this is true, if this proves to be true, then early detection could identify high-risk patients at risk for metastasis. And the cells themselves may provide new targets, which if blocked could prevent spread to distant sites. So in my talk, I'm going to talk briefly about a background on circulating tumour cells in breast cancer, because I don't think anyone's spoken about this at the meeting so far. I'm going to discuss how common they are, what is the clinical significance, can they give rise to metastasis, which has been a contentious issue for a number of years, and how do they relate to the primary breast tumour. Then I'm going to go on and describe some studies from my lab where we did a genomic analysis to try and identify unique features of the cells, and then describe where we're going with our results. So circulating tumour cells have been described a long time ago. The first report was published in 1867 in the Australian Medical Journal by Thomas Ashworth, and he found a case of cancer in which cells similar to those in the tumours were seen in the blood after a patient's death. And he felt that explained the origin of multiple metastatic tumours. They've been found in the peripheral blood of patients with many different tumour types, and I've listed a few here. And it's felt that, as I say, they can develop independent of tumour size. So even a very small cancer can be associated with circulating tumour cells. One of the problems in studying them is they're a rare population. They only make up 1 in 10 to the 7 or 10 to the 9 nucleated blood cells. And of course, most of the nucleated blood cells are white blood cells. And I've just highlighted a case here with a preparation of nucleated blood cells and highlighting a keratin stain showing a single CTC. So the clinical significance of CTCs may represent that they are linked to the primary tumour. And more conclusively, there was a publication in Nature Biotechnology in 2013 where for the first time people were able to grow CTCs in a mouse model in the femoral medullary cavity. So that, I think, proves at last that they are able to give rise to metastasis. There have been studies doing genomic profiling. This is the study that I mentioned before where they did next-generation sequencing and array CGH on colorectal cancer. And they were able to do a longitudinal analysis of the primary tumour, circulating tumour cells and metastatic sites. And they were able to identify about approximately 60% of identical alterations. And in many cases, they were able to identify specific alterations shared either between the CTCs and the primary tumour or the CTCs and metastasis. They were also able to identify unique alterations to the CTCs, and many of these were in tetraploid cells. There's been a single study published prior to ours in metastatic breast cancer where they looked at array CGH in 102 cases and were able to identify concordance of copy number profiles with the primary tumour in approximately 70% of cases, and they identified, again, a more frequent amplification of ERB2. Typically, in clinical cases, we see amplification of ERB2 in sort of 15 to 20% of cases, but in CTCs, it seems to be higher, in 30 to 40%. They were able to do serial profiling from the same patients and identified some common alterations. There are various methods to analyze CTCs. You can enrich them. You have to enrich them from the blood. You can do a venipuncture. You have to obviously do a venipuncture first. And in our study, we discarded the first 5 mLs of blood to make sure we weren't contaminating our sample with any epithelial cells from the skin. There are various ways to separate the cells. Either you can use size, you can use density, you can use immunomagnetic technology and rely on cell surface markers such as EpCAM, cytokeratin, or you can deplete CD45 positive white blood cells. There's a lot of excitement about these circulating tumour cell chips, although there are few available commercially as yet, but they do seem to yield higher numbers than the other methods. In our study, we used immunomagnetic enrichment. So we designed and validated a protocol to specifically isolate circulating tumour cells from peripheral blood, and we identified some novel alterations, which I'm going to describe. So as I say, we obtained blood samples from patients. We depleted the CD45 positive white cells. We did a cytokeratin stain and then did laser-assisted microdissection to specifically isolate the tumor cells. We then performed whole genome amplification because you get very small numbers of cells in this process. And then we did a copy number analysis using the Affymetrix SNP6 chip. And on this side, what I'm showing you is obviously when we're working with very small numbers of cells and doing whole genome amplification, there's a concern we may be introducing bias by the process of whole genome amplification. And so we did a number of studies where we compared unamplified genomic DNA, and then very small numbers of cells, and showed that this is just a heat map showing regions of amplification in the MCF7 breast cancer cell line, looking at chromosome 13, and using various small numbers of cells and whole genome amplification, we were able to generate the same profile as we would get from an amplified DNA. And this is showing chromosome 17 from SKBR3 cells where the ERB2 amplicon is amplified. So these were our sort of control or validation experiments. This is our patient population. We obtained, out of 38 patients with locally advanced breast cancer, we obtained circulating tumor cells from 53% of them. And in 17 patients, we had both tumor and normal pairs for analysis. It's hard to read, but really this is a fairly representative population of patients with advanced disease. We had 4 out of 20 patients with HER2 positive. We had 7 of our patients were triple negative, and only 5 had distant metastases at the time of diagnosis. We had collected small numbers of circulating tumor cells. In 6 of our 20 cases, we had less than 5. The first thing we looked at was HER2 status, because we knew 4 of the patients were positive in their primary tumor, and we were able to demonstrate in the circulating tumor cells. And as we had expected, we identified more patients that were HER2 positive. We did cluster analysis. This I'm not going to dwell on. I think everybody's familiar with the principle of clustering, where you do a genomic analysis of your samples, and then you group them together in terms of how similar they are, and you continue to match them until they fall into distinct groups. This is the cluster analysis for our patients, for our 20 samples that we analyzed. So cluster 1, it was a smaller subset, was only three specimens, and was fairly heterogeneous. Cluster 2 was more homogeneous. We've had 14 cases fell into that cluster, and these were more frequent. The patients with distant metastasis fell into this cluster. We found much more genomic alterations in this. So when we looked at the 2 groups, they seemed to, and looked at the genes that were altered in them, the smaller group, the more homogeneous group, composed of only 3 samples, seemed to be genes that were more related to dormancy, whereas the other group seemed more aggressive genes, and I've listed some of them here. And we also identified a common signature of genes that were altered in all the samples. When we looked at all the alterations we found, this is combining all cases, all the 17 tumor and normal pairs. We looked for minimal common regions of gain in at least 4 samples, and of that we found 414 MCRs, minimal common regions. And of these, in our recurrent signature, we identified 90 recurrent alterations that were present in 15 out of 17 of our samples. And interestingly, of the 90, 48 out of 90 were present on chromosome 19. And we had 1 MCR that was altered in 16 out of 17 of our samples. I've listed here some of the more frequent alterations, and this is just showing you the chromosomes going from 1 to X, and the regions of green are showing gain, the blue is showing loss. And as I say, this is chromosome 19, which was the most striking and most surprising finding that we had. Because we've been studying in my lab, we've been doing a lot of array CGH on many types of breast lesions, and chromosome 19 is not very frequently altered. So we went to the Cancer Genome Atlas, and we found that CTC-like MCRs that we identified were only present in 3 to 4% of samples. And I'm showing you some of the more frequently altered regions on chromosome 19 here. So are the cells that give rise to metastasis a distinct subset within a primary tumor? They seem to be different. And we know breast cancers are heterogeneous. I'm a pathologist. I spend a lot of time looking down the microscope at breast cancers. And these are 3 different breast tumors. They look quite different down the microscope. They stain differently for HER2 by IHC. And this is HER2 FISH. And again, the results are often heterogeneous. They're not positive throughout, typically. So intratumor heterogeneity is becoming very topical. It's been highlighted by next-generation sequencing. It has implications for treatment, obviously, because there's a lot of variation of the cells within a tumor. It's been appreciated by pathologists for a long time in assessing receptor status of HER2 and also ER and PR. There have been recent efforts to develop a standardized terminology to describe heterogeneity in HER2, and we really don't have good ways to describe this and good terms to communicate what we're seeing to each other, and that needs to change. And with increasing use of neoadjuvant chemotherapy, we need a better understanding of how representative a core biopsy is. If a breast tumor is heterogeneous and all we have of a tumor is a core biopsy and then they get a pathologic complete response, are we really left with a good picture of what the tumor was initially? Also, we need to understand the spatial location and relationship of tumor cells, and as I say, because we don't have good ways to describe this at this time. I think this could pave the way for molecular imaging techniques that would more accurately reflect response to treatment as well. I'm not going to dwell on this because I'm probably running out of time, but this is an interesting study that was published in 2012 in the New England Journal of Medicine. They were looking at renal carcinomas. They looked at multiple sites within a single renal carcinoma and also multiple metastatic sites. And they did next-generation sequencing and compared the mutations present. And not surprisingly, there was a lot of heterogeneity within the various sites, and they put them together and made a sort of phylogenetic relationship of the tumor types. And it seems as if not all metastases came from the primary tumor. Some of them seem to have seeded from other metastatic sites. In my own group, this is my last slide of what we're doing, we've become interested in these regions of chromosome 19. We have tumor samples on many of the patients that we have CTCs, so we're going back into the primary tumors and looking for these chromosome 19 alterations. We're expecting them to be present in a low percentage of the cells within the tumor, and our initial studies, we have some preliminary data which supports this. But we're interested to try and localize where are these cells? Are they near blood vessels? Are they at the periphery? Are they at the center? At the moment, we're just trying to work this out. The other thing we're doing is we're looking at the genes that we identified, and we're looking at a high-throughput metastasis model. This is a chick chorioallantoic membrane model, and I have a collaborator in Edmonton that we're working with. And so it's a way of doing high-throughput screening to look for metastatic potential in the tumor cells. So this is the two things we're working on. So anyway, to sum up what I've talked about the circulating tumor cells. I've given you a brief overview of their prognostic implications and metastatic potential. In terms of my own genomic analysis, we've identified chromosomal regions of recurrent gain that appear characteristic of CTCs from breast cancer patients. We've identified two groups with sort of dormant and aggressive features, and we've identified some common regions of alterations. And as I say, particularly chromosome 19 came out of our studies. The next step we're pursuing is to try and look more in depth at intratumoral heterogeneity and trying to work with the challenge of how to describe it. I think ultimately we're going to need to work with mathematicians in a way to sort of model this and develop ways that we can, standardized ways so we can compare studies and we can more quantitatively measure what we know. I mean, at the moment, just to describe something as heterogeneous is not enough, I don't think. I'd like to acknowledge my collaborators, which include medical oncologists and surgeons, statisticians, and some of the other breast pathologists I work with. These are my lab members who worked on it, particularly Nisha Kanwar. She provided most of the data. These are the funders of the work, and I'd like to thank you for your attention.